All right, I want to welcome all of you to our PolarConnect event for this bright and sunny day here in Alaska. It's August 31st, 2016. This is Janet. I work for the Arctic Research Consortium of the United States, and I'll be facilitating this presentation today. Also joining me is Judy from ARCIS, and if you have any technical issues as we go along, feel free to type them in the chat area, and Judy and I will try to address them as we go along. Our presentation today is features Nell Kemp and Rebecca Hewitt, and I don't know if the other researcher, Michelle Mack, is with them today, but they're going to be talking about some research they've been doing up at Toolik Field Station and near Healy, Alaska. We're pretty excited to hear what they've been up to and get a perspective of Nell's experience in the Arctic. Before I turn the presentation over to the ladies in the webcam, we have a few things that we want to go over with this platform. It's called Adobe Connect, and if you haven't used it before, the presentation will be showing up in the center of your screen. Many of you have discovered the public chat area and are saying hello, and this is where we will also post your questions, or if you have questions, this is where you'll type them, and then we'll relay them to the team. There is a microphone that might be showing up above the Adobe Connect slide. Please click on that to mute the microphone. If you've joined us by phone, *6 to mute your phone. We'll also mute any background noise so we get as best of presentation as we can. We'll see how the webcam does for Becky and Nell. Sometimes if we have bandwidth issues, we'll have them shut it down in case it's getting too weird and we get frozen heads and things like that. This event will be archived, and we'll send out the link afterwards and post it in a couple of days on the website. Again, before I turn it over to them, just a little bit about the program and why Nell is with a researcher team. She is part of the PolarTREC program, and there's a lot of information on our website at PolarTREC.com. But basically, we have been placing teachers with scientists around the polar regions, both the Arctic and the Antarctic, since 2004. The program is administered by a nonprofit, and we're based in Fairbanks, Alaska, with an office in D.C. And we do a lot of other things, but this is one of our better and more fun programs where we put teachers out with researchers so that they can get hands-on experiences and take that back to the classroom and become better at teaching science. Nell is not new to this program. She's actually an alumni and has been to Toolik before, which I'm sure she'll comment on. But we're really excited to have her again working with the research team and doing great things. After their presentation and as we go along, I'm sure you'll have some questions. So during the presentation, please type them in the chat area, and since Nell and Becky are both there, they probably can see that chat and respond on the fly. If not, we will relay the questions to them and kind of remind them to answer those as we go along. And then at the end, we'll also put the slide back up, and you'll be free to ask your questions live if you don't want to be in the chat area. And I think that's it. So for my end, I'm going to turn this over to Nell and Becky.
Well, welcome everybody. We're really excited to have you here today. As Janet mentioned, this is my second time with PolarTREC. I was very fortunate to go in 2013 to Toolik Field Station, and I was really excited that I got to go back. If you were reading my journals, you will notice one of the things that I commented on was that I went to Toolik in July the last time, and just being there in 2 or 3 weeks later, the colors had changed for the fall, so the tundra was really, really beautiful this time. So this picture right here is the boardwalk out to our field sites, and so you can kind of see the yellows and reds of the changing tundra. Okay, so The name of this project, the full name, Interrupting the Deep Freeze, Below Ground Exploration of a New Environment as Permafrost Soils Thaw. Our PolarTREC name is Deep Roots because that's basically what we've been studying for the past 2 weeks, and Becky for the past year and a half. So actually what this study is talking about is basically figuring out, I'll go through the whole thing, but figuring out how the roots that are deep in the tundra soil are going to react as the climate changes, and the permafrost thaws, and whether or not they'll be able to access deep nitrogen. So this is Becky and myself. So Becky is actually injecting some nitrogen into the permafrost soil, and I'm holding back plants. We joked that during this experience, my yoga came in handy because there were a lot of times we had to hold back plants to make sure no nitrogen tracer got on any of the plant material. Okay, so basically these are the research questions that the Deep Roots team is investigating. So again, can plants that are in this tundra environment access that very deep nitrogen that is stored in the permafrost layer? And a couple of things about accessing that nitrogen. So there are a couple questions about whether or not it's just the traits of the roots themselves, how deep they are, what plant they're coming from, but also what relationship they have with special fungus called mycorrhizae. Yeah. So to see if those fungus are going to allow them to reach that deep nitrogen. So the other question was, if these plants can access this deep nitrogen, where does that nitrogen go? So where does it go into these these tissues of the plants? Do they go to the leaves? Does it go just in the roots? And Becky and the team was looking at a 24-hour period and a 1-year period to see where that nitrogen ended up. And then lastly, and we'll get to this part in a couple of slides, but basically the larger question here, and I always call it the elephant in the room, is the fact that our climate is changing, our Arctic is warming, and when that happens, more carbon dioxide and methane is released into the atmosphere. And so if plants can access this nitrogen and grow a lot bigger, potentially they could photosynthesize at a higher rate, take in some of that carbon dioxide, and perhaps slow some of the warming that's happening in the Arctic. So our research study, we actually have 2 locations. The first, which we're going to tomorrow, is 8 Mile Lake, which is in Healy, Alaska, right near Denali National Park. So that's just southwest of Fairbanks. And then our other research site was Toolik, which is, I think it's 60 miles north of the Arctic Circle? No, more than that.
I don't know. Miles
goes
284.
Far north of the Arctic Circle, just north of the Brooks Range in Alaska. Open tundra, no trees, beautiful environment. And if you look in this slide, you can see one of the big things that's different between Healy, Alaska and Toolik is the permafrost layer. So there's something called continuous permafrost, which basically just means that all of the soil has this permafrost layer. And permafrost is something that has been frozen for 2 or more years consecutively. Whereas in the warmer climates of Alaska, down south near Fairbanks and Healy, you get discontinuous permafrost. So you get some areas of the soil where permafrost exists and some doesn't. So in 8 Mile Lake, you have more trees, and you have more shrubs. So the root profile can be a little bit different. So you can see the permafrost is a lot lower down than it is in Toolik. Okay, so permafrost. So I like to tell my students that permafrost is Earth's freezer, because essentially what happens to the permafrost is that this is a permanently frozen layer of soil, and it can be frozen for minimum 2 years to up to 20,000 years. So what happens is you get a lot of vegetation and animal material that has never decayed or decomposed. That's just sort of locked in this freezer. So it's just sitting there waiting to be waiting to decay. So what that means is that there's potentially a lot of nitrogen and a lot of carbon that is stored in this permafrost. And so as our climate is warming, this permafrost is thawing. And when this permafrost is thawing, that means all these little decomposers and detritivores are going to come into that dead material and try and break it down. And when they break it down, they release carbon dioxide, they release methane, which, as we know, are greenhouse gases. So that is what we call a positive feedback loop. Basically, more plant material breaks down from that permafrost, more carbon dioxide is released, our climate warms. And so you can see in this slide, this is just sort of a nice little diagram showing you that cycle, but you can also see that the permafrost is covering a large portion of the northern part of the Earth, like the North Pole, we like to call it. So a lot of upper Russia, a lot of upper Greenland, upper Alaska and Canada is being covered by this permafrost. Sorry, we just had a little interruption by the UPS guy. So why does the Arctic matter? I kind of said it already, but the Arctic is warming faster than any portion of the Earth. So as this Arctic warms, the permafrost is warming as well. So the other big question that the Deep Roots team is investigating is nitrogen. And if any of you are home gardeners, you probably know what nitrogen does to your plants, right? So I've included a nice little picture of fertilizer here to remind everyone that plants really, really, really love nitrogen, just like we do. Our DNA is made of nitrogen, our proteins are made of nitrogen. So basically, if you're adding nitrogen to soil, plants are going to take it up and they're going to grow bigger. They're going to be able to build more structures. And because the tundra soil has this permafrost layer underneath, which has been frozen for so long, the plants are very limited in the amount of nitrogen that's available to them. So the question sort of becomes, well, you know, if the permafrost really is warming and this nitrogen is going to become available as this permafrost thaws, will the plants in the tundra be able to access this nitrogen? And if so, what will that do to them? Well, where will that nitrogen end up? Will these plants grow bigger? And if they do grow bigger, will they photosynthesize more? Will they take in that carbon dioxide? And perhaps they will help to sort of modulate that positive feedback cycle that's predicted to occur. One of the really interesting things I found working in this project is the fact that the Deep Roots team doesn't just consist of Dr. Hewitt and Dr. Mack, because there are, what do you call them, collaborators? Yeah, collaborators from various institutions who have sort of secondary questions that depend a lot on the research that the team is doing up at Toolik. So for example, one of the things that we did, and you'll see it in the future slide when I go through some of the pictures, one of the things that we did was to evaluate various different types of tundra all along the Dalton Highway. And what this allowed the researchers to learn is to learn sort of the different types of tundra,
shrubby versus
raminoid-dominated tundra. Raminoid-dominated tundra. Rocky, glaciated, peaty, all these different types of tundra that exist. And it allows, there are two scientists here at the University of Alaska Fairbanks that are climate modelers. And so what they want to know is they're going to take this research from the Deep Roots team, look at these different areas of tundra, and sort of try to make a model or a prediction about what could potentially happen if our climate warms. So if they know how these plants take in this nitrogen and what they do, and they sort of figure out what plants exist in these different areas of tundra and what the thaw depths are and all these different pieces, they can then go back and then make a predictive model so that scientists can then figure out, well, if this really does happen, here are some things that we can do to sort of, I guess, react to it. Another scientist who was on the team was actually very interested in the fungus, the mycorrhizae that exists around these roots. So he is very interested to figure out, and we'll talk a little bit more about this later, but what fungus exists in the tundra, and do, different fungal communities give roots sort of a different edge in different areas of the tundra? Does this allow them to take up more nitrogen? So it's really interesting for me as a teacher to sort of see how all these different scientists from different institutions sort of are getting at the same larger question, which, has to do with climate change, but they all have very different interests. But they also, are 100% collaborators. Like the conversations, and I think I wrote a journal post about this, the conversations that were happening between them were something that I think 20 years ago when I was in university, I didn't think happened between scientists. It seemed like everyone was sort of in their own lab and didn't talk to each other. So it was really nice for me to see how these scientists communicated, because that's what I always tell my students, is that, science is a communal enterprise. It's not just an individual in their lab. So that was really neat for me to see. Anyway, so as I mentioned, one of the scientists is interested in just the fungal communities. And so this is a slide that Becky created for another presentation, but it just kind of gives you an idea of these fungi throw down these sort of tubes at the ends of roots, and they can extend the roots to huge depths. And when we were investigating the permafrost layer, the thaw depth in some of these plots could be as much as, I think, the 85 centimeters. Yeah, what's the greatest depth. Mm-hmm. So you can imagine that a plant root, would have to be extremely long, 85 centimeters. So the fungi kind of give them a little boost. They can extend those roots a little bit. And they extend them not just in their depth, but they sort of create this wide network. If any of you listen to Radiolab, I'm actually going to put this in my next journal post, but they did a really interesting piece a couple weeks ago about these fungal communities in the soil. It was really, really neat. Just miles and miles and miles of fungus underneath our feet. So this is what you really want to see. These are our pictures from out in the field. So in the upper left, I guess it is, that's Becky in the blue jacket, and that's Dr. Lee Taylor. So he is the mycologist. He's interested in the fungal communities. So they're just out on actually this was just a random tundra site on the Dalton Highway, so that they were scoping out for next year. And right below them is Dr. Helen Jeanette. She's here at University of Alaska Fairbanks. She's actually cutting out a soil monolith. So they were just cutting out soil to sort of get an idea of, one, how great the thaw depth was in these different communities, or these different tundra plots, but also to see what the soil profile was like, to see whether it was rocky, whether it was non-rocky, how waterlogged it was, etc. And these, again, are the sites that they're thinking about using for next year, for maybe doing some modeling. The pictures on the right are all pictures of roots. I know, it's really shocking. The one on the bottom right is sort of the team's favorite photo. And it's a favorite photo because it's an extremely close-up shot of the roots, and it's showing all of those little fungal tips. And if you saw them in real life, they were like bright orange. They were really, really neat to see. And they're very tiny. I happened to go on Amazon and buy a little magnifying macro lens for my camera. So yeah, so that's pretty neat. So basically that's a huge part of the experiment, and you'll see it in later slides. But so when we were working at Toolik, we had 8 plots. So we had 4 plots in greenhouses and 4 plots in non-greenhouses. They were our control. And basically we wanted to see, do these roots act differently in a warmed environment versus just a traditional tundra environment. So you can see that in the first picture in the upper left, we're placing out a quadrant so that Becky can inject, the next day she's going to inject nitrogen into each of those flag markers. And so what she did was basically, I think it was every 2 centimeters, right?
No, they were 25 centimeters.
25 centimeters, sorry. So each flag is about 25 centimeters apart, and there were 13 injection points. And so each of those flags, the next day, you'll see another picture, will be injected with an isotope of nitrogen so that we can trace it through the plant in 24 hours. And then again, there's a secondary plot, which there's not a picture of, that will be harvested in a year. And so then they're just laying down the plot as well. You can see the picture to the right is a little more awkward, the quadrant they're laying down, because they're laying it down in a greenhouse. So that structure right there is the greenhouse. All the plastic film's been removed. But the plant community is a little bit different in there because this greenhouse has existed for something like 20 or 30 years. So the plants tend to be a little shrubbier in that environment. And then just the pictures below are just sort of, again, cutting out soil monoliths just to get a profile of what the soil looks like, what the roots look like, and just sort of what's happening below ground. Okay. So this was one of our biggest work days. This was actually the next day. So each of those flags, there's sort of this metal cross, I guess is the best way you could describe it. But they're injection needles. And so what we would do is we would go out to each of these plots. We would take a frost probe and find the thaw depth and sort of wallow out a little hole, and then stick this injection needle in. So these injection needles were made by this awesome machine shop here in Fairbanks, to Becky's specifications, and Michelle's, yes. They're really neat because at the very bottom of them, they have tiny little holes which will release the nitrogen tracer. But those holes need to remain covered essentially until they're all the way down at that permafrost layer. Because you don't want any of the nitrogen tracer to sort of get into any area except right at thaw depth. So basically you plunge them in, reveal the tiny holes, and then you can see in the upper right corner, Becky is taking a syringe full of ammonium chloride, and the nitrogen again is an isotope, of nitrogen. It has 15 neutrons instead of 14, right?
It's 15 total neutrons, yeah.
But it's normally 14.
It's normally 14.
Yeah. Anyway, so then we are injecting the syringe into each needle. And you can see in the picture on the right, the reason that I'm holding back all of the foliage is because, again, we only want that tracer to end up very, very deep in the permafrost so that we can trace where it goes in those plants if they can take it up. Okay. So this is a diagram basically just showing exactly what I talked about. So you have the needle that is very low in the ground at the permafrost level. And so we have 8 plots. The syringes basically are going to inject that tracer way, way down so that we can see if the roots will take up that nitrogen. Yeah. Oh, and I should mention, I think I mentioned this once before, but we do have 2 plots at each of these 8 replicate pots. So we have one that's harvested 24 hours later after the injection, and one that's harvested a year later. So we inject all of the plots, which was a very long day in the field. I think we were out from 8 a.m. till 10 p.m.
A few days,
yeah. And then the next day, a team came in. We had actually, I would say, 8 to 10 people working on the team at this point.
Yeah, some in the field still, and then about 8 to 10 people plucking at that point, yeah.
So the pluck is the next big thing. So basically, after 24 hours, we want to figure out where that nitrogen has ended up. So several things happen. You have, if you look in the sort of lower left corner, you can see that people are dissecting a soil monolith for the above ground. So they're trying to see all of the plants in this harvest plot. They're separating it into the different species of plants that exist, and then they further separate it into tissue types. So for example, new stems versus old stems, new stems versus old or new leaves versus old leaves, etc. And those all get bagged. You can see there's a bunch of pictures of sample bags. Those all get bagged and tagged. They get unique identifiers because they're going to go for further analysis. There's two different analyses that happen. One is isotope analysis to see if the nitrogen ended up in any of these plant tissues, if they ended up in new leaves, in old leaves, etc. And then the other is for RNA, RNA.
DNA and
DNA
RNA
and
when
RNA.
we're down on the roots in the
Right,
soil.
which is another part. We'll get to that in a second. So if you look at the sort of big tundra brownie, as we call it, in the upper left, that is a block of soil that was cut out from each of our plots. And so what has to happen is what we call the pluck, which means a bunch of people sit there and pluck out every single piece of plant. And as I said, they put it into categories by a species type. But then they also are going to take every single root. And so there are a bunch of different samples that get cut out. There are some where the roots are still intact with their plant above. And then there are some where it's a little bit lower down and you just have a mass of roots and you just look at roots and try to figure out what species they belong to. And so those roots that get identified much deeper down are separated into their species as best we can by eye. And then you'll see in the middle lower picture there's someone looking at a microscope. Those roots are then identified under microscope. And just like any other species of animal, each species of plant has distinct characteristics. That when people look at roots, they can figure out this is a Betuliniana or this is a Salix pulchra, even though those are the two that are
very hard to tell.
They're very hard to tell apart. So under the microscope, someone who is much better at this than me basically separated them into their different types. And then what we did is we took each of those, they were scanned on a very special root scanner, and they were, a digital copy was made, obviously, from the scanner to get the root length, and also the root length was taken. They were then put into sample bags. You can see that in sort of the upper right-hand picture. So if you were to zoom in on that picture, you would see that there is a species name for that root, and there's also a unique identifying number. And that number tells whether that root will actually go off for further isotope analysis to see where the nitrogen ended up, or they will go for genetic analysis. And the genetic analysis is basically twofold. One is to make sure that, yes, that is actually the correct species of root. But more importantly, it's also to see what fungi are living on those roots. So we want to see through genetic analysis what are the different fungi living on those roots, and then sort of see if perhaps there's a correlation between the amount of nitrogen they can take up and the fungi living on those roots. Right? Okay. All right. And then, we're almost done. So then this last slide is just about the fact that we're going to Healy, Alaska, actually tomorrow, and again this weekend, because we're going to do the 1-year harvest. So Becky was here a year ago, last summer, and she did that same injection in Healy, Alaska, and they harvested it 1 day later. But now it's time for her to do the 1-year harvest. And this harvest is a little bit trickier because the soil is a little bit different. It's a little bit warmer, it's a little bit wetter, it's a little bit sloppier. As I said to my students, it's sort of like imagine cutting an ice cream cake. It's actually easier to cut when the ice cream is very hard and very frozen, because they can just sort of take a nice slice out of it. But as the ice cream starts to sort of melt, then it gets soupy and you can't get a nice clean cut. So we're hoping, because it's supposed to be all sunny and beautiful this whole week, that our coring will go without many problems this weekend. These are pictures of some of the sampling that was done next week. I'll probably, I would assume, be writing a journal about the cypre core and how that works. Basically, you just take a big soil core and permafrost core, sort of like a huge hole punch, but it involves a drill and a lot of labor. It should be really interesting, and hopefully we'll see some nice pictures, or you'll see some nice pictures, and we'll have nice weather to see Denali. yeah, so that's happening tomorrow. And that, I think, is pretty much it. Did I leave anything out?
Nope, I think you covered it.
Okay, well, thank you.
Yeah. Yeah, and there were, nice job, ladies, and while you were chatting away there were several questions, so showing up in the chat area. So let me go back to them. Oh, well, my question was, since you started listing all those tundra types, how many different types of tundra are there?
Well, we're focusing on 4 different surface ages. So as you come out of the Brooks Range and move across the foothills to the north, we're in glaciated surfaces that are either rocky or peaty in terms of their substrates. And then as you move north towards the coastal plain, you can enter areas that have not been glaciated recently or unglaciated but are still rocky, and then there are also silty surfaces. So we're trying to understand whether these different soil formation processes, glacial history, surface age, those sorts of landscape scale factors influence rooting depth profiles and nitrogen availability. So we're focused on those 4 types, ecosystem types or landscape types.
All right, cool. Okay, so Josh had a question, and we might have to find a photo that goes with this, but how abundant are plants, legumes with symbiotic nitrogen-fixing bacteria in these tundra environments?
So there aren't that many legumes, although there are some invasive species that you can find along the haul roads further and further north each year, it seems, and also down riparian corridors. But we do have a nitrogen-fixing shrub that occurs in, as far north as we were researching this summer, but it is not abundant in our plots. But it does exist on the landscape.
Okay. Let's see, lots of comments about, I love these photos to the roots, a lot of details to pay attention to. Let's see, Josh again, once a sample is collected, is anything done to stop the metabolic activities of the organisms in the sample that would be moving the nitrogen around between organisms and tissues?
Well, for the RNA and DNA analysis, we put samples immediately into a highly, highly concentrated salt solution that basically locks everyone in place so that you can extract high quality DNA later. So we put that in this very strong preservative. For the RNA work, we put samples immediately into liquid nitrogen. So with those 2 sample types, everything is just sort of locked in place and preserved. And otherwise, we're sampling plant tissue and looking at sort of bulk isotope occurrence either in soils or in roots that are fairly well cleaned up. Yeah, I don't know if that answers the question fully.
All right. Bruce H. said, thanks for the interesting presentation. He has to leave. And he said, incredibly detailed work, micro-plucking using the genetic info is such a good idea, huge improvement. Thanks, Bruce. Let me go back up here. I had a question actually about this photo that you already have, the one that has the detail. I might have missed this when you were talking, Nell, but what is the scale on those photos? I assume they're fairly close. But
I feel like it was 20 times magnified.
I don't know. I don't know what the magnification was. I never
yeah.
saw that. But those root tips are probably the bottom photo that Nell was pointing out, where you can really see the highly infected ectomycorrhiza. Yeah, that one. That was probably about a centimeter or centimeter and a half for the full extent of that colonized region.
wow.
But it's so exciting because you're sitting there picking roots, and then you see something that's hot pink, and you know it's ectomycorrhizal in it.
Yeah.
Yeah.
As far as root plucks go, it was pretty exciting.
That's great. Does anybody have questions they want to ask live? Let's see. I'm looking to see what's going on here. So yeah, Judy was talking about the field site. I want to know now, you know, like how did this experience compare to your first one? And, you know, was it not so much maybe the science, but just the whole aspect of going back out in the field and working with teachers or with researchers, and just kind of what your thoughts were about the whole second time?
The biggest difference was the fact that there were no mosquitoes, which was very, very nice for me. You know, it was really, really different because the first experience I had a very small research team. It was just myself and two others. It was just the PI and a research assistant and myself. Whereas this one, we had so many different scientists, research assistants. We had such a huge team. So that was a huge difference. And like I said, just being able to see that collaboration across universities and across disciplines, you know, so that we had people who were really just there for genetic analysis. We had people who were really into the fungus. We had people, you know, just all these. And that was really neat to hear these conversations that were happening amongst scientists. As far as, you know, going back to Toolik, it's like going back to summer camp. Like everything is kind of exactly the same, but you know the people have changed, but the routines are kind of the same. Food's still really, really good. We were much more concentrated at Toolik at the field station, whereas in my last experience, we drove out along the Dalton almost twice a week to our different field locations. Yeah, I mean, it was very different, but very much the same, and I still loved it. It's still as beautiful as always. It was really, really neat to be there at different times just to see the colors.
Okay, yeah, yeah, it's a pretty time of year up there for sure. Looks like people are typing questions while they're doing that. I want to ask Becky because I think this is the first time, at least for your team, that you've had to work with a teacher. Kind of what is it given to your project, or you know, what are your thoughts about having an educator as part of your team?
Yeah, so this is my first time with PolarTREC. Michelle, who's the PI on the project, has hosted a PolarTREC teacher before. But for me, it was just sort of provided an injection of enthusiasm and just a really different viewpoint on our research. It was really great the first few days having Nell ask these fairly basic questions about the project, and just, you know, I would answer them, and then the follow-up question really provided an opportunity for me to clarify and hone in our message of what we're trying to do on the project, and then to see how she could take that and then translate it even further to what would be accessible to her middle school students. And it was so exciting every day to hear, and very impressive, the questions that her students came up with based on her journal posts. First of all, that they were interested in following our project during their summer vacation was, you know, speaks huge amounts of her as an educator in the first place. But they just came up with the most fantastic questions. And I mean, they really were questions that sort of represent the cutting edge of what we're trying to figure out within this topic of research. So I guess it just provided quite an energy boost and was an exciting facet to the research team up there, but also has really affected the way that hopefully in the future I'll be able to communicate about this project. And is just a first step towards, you know, we started to talk about, okay, well, how would we continue to work together in the fall, and what would be meaningful for our research team at NAU, and then Nell as a teacher, and then her students. And that's an exciting way to move forward, I think.
Yeah, I will say I was extremely shocked that my students were like engaging in my journals and asking so many questions because it is their summer break. And I had, told them in June they needed to read my journals, but I really did not expect so many of them to make comments. And the questions that they were asking, I mean, it just showed to me that they cared about what I was doing. They had paid attention last year in environmental science, and they were actually reading the journals and making very thoughtful questions. And some of the questions that they asked, we even couldn't answer. They were asking, one of my students asked a question about like, well, if you guys are collecting so much evidence, how come people still really don't believe that this is happening? Like, what's wrong with people kind of thing, these philosophical questions. But having the students, ask questions about fungi, like that was so exciting to us. They asked questions about these ericoid plants. I mean, it was amazing. And so I think it got the research team really excited that, Becky said to me at one point, she said, sometimes you can get so deep in the research, you just sort of forget why you're doing it. And you, sorry, I'm like going out of frame here. Oh, there we go. She said, you forget why you're doing it and you feel like you're not really doing any good. You're just sort of sitting in a lab. And so I said that one of the great things about PolarTREC is that it allows researchers to see that what they're doing really does affect students, that they are following along, that they are thinking about how they could possibly be a scientist in the future, or how their research can inform them when they're talking to their parents about science or climate change, or when they're voting or anything like that, just keeping their minds open by paying attention to this research. And I think that they're now obsessed with this project, and they would absolutely love meeting the team in person. We've actually already talked about, if we could replicate this experiment on a much smaller scale with my students in the classroom, that would be really fun for them.
Oh, yeah.
And I know someone wrote a question in here about how am I going to use this with my students. And, there was someone here from the National Science Foundation. She was up at Toolik, and she was asking me kind of the same question. And, the thing I said about PolarTREC is that I can't tell you, like, what lesson I'm going to create or what specifically I will do with this experience, but I can tell you that as an educator, it informs every decision I make in my classroom. Every time I make an analogy about anything, I often refer back to my PolarTREC experience. I talk about animal adaptations to the Arctic when we're talking about adaptations and evolution. A lot of my students were asking questions about some of the plants and animals in the Arctic, and I said, well, I'm so glad you're asking this because our first unit is evolution, and this is exactly what we're going to talk about. I find that this experience comes up at least once a week in my classroom, and that was from my 2013 experience. So I can't even imagine having a secondary experience, what this will do for my teaching and my students.
Oh, yes. I'm going to use all of your quotes now. No. Thank you. No, and I think you did a good job on asking Kara's question about using in the classroom. Let's see. There was a question above that from Marie June, I believe. How do you remove the plant without cutting the roots? So kind of back to the science there, and thank you.
How can you? We do cut the roots. So, but what we know then is the amount of roots on a per area basis. So, yeah, I don't see her question.
It was just, it was up above. It's okay. It just says, how do you remove the plant without cutting the roots? So,
yeah, so we saw right in, and we purposefully have to cut roots, but then we know the exact content within that volume of soil of these large coarse roots, or the fine root biomass. The fine roots are the ones that are acquiring nutrients and water. so then we can make our estimates of the actual biomass of those types of roots within that volume of soil.
The same person, Marie June, asked, any suggestions on how I can use this research in my 9th grade environmental science class at their level? I hope you can share some resources, and your research is awesome.
That's a really good question. Actually, you know, I am teaching biology this year, but I do generally teach 7th grade environmental science. And that was one of the questions Becky and I had, was whether or not we could develop a lesson basically doing the same kind of thing, like injecting maybe a dye or something deep into different soils to see how plants take it up. You know, I was using the example of like when you were a kid and you put carnations in different food coloring and you saw the carnations change. But maybe trying to do something like that to help them understand, A, that plants are going to take up whatever is in the soil. And then that's pretty much at this point as far as we've gotten. But definitely it is in the works. I would say that, you know, using my journals would probably help your students a lot with sort of understanding, you know, this was a really complex project. And so I really had to think about how to write journals. You'll notice, I think my journals, I split the experiment into like 4 or 5 different journals because there's sort of a lot of background knowledge that students need before they can understand this experiment altogether. So I think helping them understand basically the role that plants in the Arctic have with, you know, greenhouse gases like carbon dioxide is one thing. Understanding permafrost is another thing. Understanding that all of these nutrients that are in the soil are limiting the growth of certain plants. You know, all of those pieces sort of move together. And so I think that you can approach this in a lot of different ways in small chunks like that, and then sort of maybe talk about this experiment sort of at the end, sort of synthesizing all those pieces. My students, part of the reason why their questions were so good is that they had already had all this knowledge last year. So they got to that point very quickly. You know, when they started reading, they said, oh yeah, basically want to, you know, slow down that positive feedback loop, which I was really impressed that they got there that quickly. But yeah, there's I don't know, there's, you know, besides my project, PolarTREC has a ton of lessons and resources for using in your classroom. I know somebody who was a teacher who was there in 2013 with me, Bruce Tatterka. He developed for his AP Environmental Science class all these different experiments based on his work at Toolik, which also had to do with nutrient cycling in watersheds. I think it was like nitrogen and carbon. I don't really remember. But those are some good resources to check out as well.
Yeah, and there is a lot. There's several people that have, well, we've supported several researchers that are doing work up at Toolik and by Healy for multiple years that have had teachers looking at that whole carbon balance kind of a thing. And I just actually posted a journal from another teacher that was at Healy this summer, but she worked with the Deep Roots team and did a little bit of sampling and wrote a journal just before Nell got there. So I posted her version of looking at that same research project there from Karen. But lots of good resources there, and of course the university's web pages as well have some probably, you know, more details if you need. And I'm sure the research team and Nell will be available also for questions or, you know, feedback or help if you have anything that you need help with. All right, let me see. Anybody else have questions there? I was looking up stuff, so nothing. Okay. Oh, I had a question for you about kind of post-field analysis. So I'll find the picture at the same time. But you said there was, you know, things were going to be analyzed back at the lab. My first question is actually, how long did it take you to process these samples that we see in the slide right here and get them ready for post-production? It's just kind of a time frame thing because didn't have a sense of that. And then what do you do and how long does it take to analyze all of this? Like, what's the big scale we're looking at?
Sure. So this took us about a week of plucking with everyone working very long hours. I guess probably upwards of 14 hours a day for a crew of about 12 people. So that was for our 8 plots at Toolik. And last year it took us about 2 weeks to pluck the full 10 plots from Healy, so we're anticipating that as the timeline just to move through the steps that Nell described here. And then it takes almost the rest of the year to separate samples further. So part of the above ground pluck is clipping new tissues from old tissues, but then we get back to the lab and we individually pluck off new leaves from new stems so that we can determine where the isotope tracer has ended up. All of the above ground and below ground parts of these plants end up getting ground into a very fine powder, and then they're run for isotope analysis. And so that's something that all of that happens back in Michelle's lab, and she's got a really great model for recruiting undergraduate researchers from the university. And so through our email exchange, it sounds like we have a slew of 5 to 10 undergraduates that will be hired in the fall, a portion of which will be working on this project, doing the separating and the grinding and what we call rolling, or scooping up small amounts of that fine ground powder from the root and above ground samples into these little tins that then are combusted in the mass spectrometer that gives us the isotope data. So it takes several months, and this will be a big year because last year, we were just starting this project at Healy, and I'd say all the samples were prepped by the spring. But this year, of course, we've got our project at Toolik and at Healy, so I imagine we'll be very busy grinding and running isotope analyses probably right up until the field season.
All right. Yeah, sounds intense. I can't imagine that you had that many people doing all that, and if it was just only 1 or 2 of you, it would take years. Michelle Mack, the other lead researcher on this project, she was also sharing that Nell's stuff will be useful for undergraduates, and that is certainly true. So, okay, let's see. Anybody else have any other lingering questions out there? If not, we'll wrap this up here pretty soon. So now, while I'm waiting here, when do you head home?
I am leaving next Tuesday morning. Right? Yeah.
Yep. And then
6:30 flight.
And then how about you, Becky? When do you head back home?
Same time frame. Yeah. So we will collect these samples, harvest them in Healy, and then ship them all back to NAU and do our Healy Pluck at Northern Arizona University. So we're just about to get started again.
Yeah, we're very hopeful that we're going to get all the field work done by Sunday night, and then we'll have Monday to sort of clean up, pack up, and but this is not going to happen. But in case it does, we do have Monday as another day where we can go out in the field if needed. But it's not going to happen.
And then you'll follow the cranes and the geese out of state and head on down south.
Yes, exactly.
Since I know they're all showing up there in Fairbanks. All right. Well, thank you very much. Before everybody bugs out here, I'm going to switch screens on you all, and we have a poll. So please answer the 2 polling questions before you sign out. And while you're doing that, I just want to thank Nell and Becky for your time and doing this event with us. It was very interesting, and we look forward to posting it online here soon and following you over the next little bit of research time and your time in Alaska.
Thank you very much. Yeah.
All right, and then if there's anybody, I'm going to stop the recording, but if there's anybody that